Hello, I'm Dr. Jonathan Holt, physician and owner of the Harmony and Focus Wellness Clinic in Highland Park, Illinois. I am a triple board certified osteopathic physician with over 20 years of experience. I have board certifications in internal medicine, clinical genetics, and clinical informatics, which is a subspecialty of preventive medicine. Today, I'd like to review the health benefits of whole genome sequencing and how it applies to your health. As compared to the field of genetics, which refers to the study and application of single genes and single gene disorders, genomics is a term that we use which is relatively new. And what genomics describes is the study of the entirety of an individual's DNA, the entirety of their genome. So it's really just a fancy way of saying all of your DNA. In the most basic sense, you get half of your genetic information from your mother and half of it from your father. And this information encodes everything about you, from your hair color to your eye color, and even affects your health and your risk for disease. DNA sequencing has come a long way in the last 30 years. After college and before medical school, I remember doing Sanger sequencing with radioactive labeled electrophoresis gels, laboriously transcribing the A's, C's, T's, and G's on the gel with a pencil and paper and typing that into a computer. It got more advanced using lasers and capillary gels and chromatography which automated this process. And as a result, over the years, the cost of DNA sequencing has come down dramatically. What used to cost hundreds of millions of dollars for a single genome, now in 2023 only costs about a few $100s. So what is whole genome sequencing? Well, whole genome sequencing is a cutting edge technology that allows us to read an individual's entire genetic code. This includes all the DNA that makes up a person's genes, as well as the other DNA sequences in between the genes that still play an important part in health and disease. By analyzing an entire individual's genome, we can gain valuable insights into their unique genetic makeup and how it may impact their health. With today's technology, DNA sequencing has benefited tremendously from automation. Modern DNA sequencing is achieved at scale by first purifying the DNA, usually obtained from a cheek smear or a blood test, purifying it, fragmenting it into smaller pieces. Then, by mimicking replication, the DNA molecules of all the fragments are sequenced in massive parallel fashion using digital cameras and computers, and then stitched back together using computer algorithms to analyze and interpret it. This works in three basic steps of amplifying, sequencing, and analyzing the DNA sequencing. One of the most common methods of doing this is called sequencing by synthesis. Molecular adapters are attached to the fragments of DNA and loaded into wells, which are basically, or these flow cells, which are basically small little petri dishes where biochemical interactions can occur. The modified DNA is loaded into these cells, and these flow cells contain small wells or holes called nanowells that space out the fragments and help from overcrowding. Each nanowell contains the building blocks of DNA strands called oligonucleotides. These are the A's, C's, G's, and T's that provide all of the anchoring points for the adapters to attach. Once the fragments have attached, a phase called cluster generation begins. And this step makes about 1,000 copies of each fragment of DNA as it done by bridge amplification using PCR. These primers and modified nucleotides are washed onto the chip, and these nucleotides have a reverse fluorescent blocker, so the DNA polymerase can only add one nucleotide at a time into the DNA fragment. And each round of synthesis, a camera takes a picture of the bioluminescence that comes off of it, and which is later used for the interpretation. So each round, non-incorporating molecules are washed away, and a chemical blocking step is then used to remove the 3' fluorescent terminal blocking group. This process continues until the full sequence of the molecule is sequenced. Here it is replicating the entire sequence, in which case you then have two copies of the DNA as it was replicated with these reference points attached to it. So these variations that we find in the DNA that we all have include single nucleotide polymorphisms, or SNPs, also larger structural variations. And these are just variations. Some of these variations originated millions of years ago, whereas others are new variations, also called de novo mutations. Okay, so great. How is this used in healthcare? One of the most significant benefits of whole genome sequencing is its potential to identify genetic variations that may affect your health, such as increased risk of developing certain diseases, and how we could use this information to keep you disease-free. Now, I want to sidetrack just a moment and talk about the difference between genetic contribution towards health and disease. In one context, a particular variant may be beneficial to survival, whereas in another context, that same variation, also sometimes called a mutation, may be detrimental. A good example of this is in the CFTR gene, which is the gene that causes cystic fibrosis. Now, this variation isn't that uncommon. In fact, about 1 in 20 Northern Europeans have a variation in this gene, and typically that person isn't affected with cystic fibrosis. It's only when that person has a child together with someone else who's a carrier for that they together have a 1 in 4 chance of having a child with cystic fibrosis. It's interesting that it is believed that one of the most common variations in the CFTR gene, called Delta 508, originated in the 13th, 14th century when cholera was an epidemic in Europe. Turns out being a carrier for this particular variation in the CFTR gene conveyed a survival benefit, as a carrier didn't die as often from cholera as people who didn't have this variation, and hence people had a survival benefit. They tended to live and have children, and this was called a founding mutation, and it's now not that uncommon in the general population. It's only a problem when you actually, as I mentioned, if you have a child with someone else who's a carrier, and this happens about 1 in 3,600 births. So by understanding this context and recognizing how genetic variations can contribute towards health and disease, individuals armed with the right information can take proactive measures to manage their health and mitigate their risks by taking preventive strategies before disease even starts. And the parable of the Taoist farmer is probably apropos, where there isn't any good genes and bad genes. They're just genes and variations. Sometimes they benefit us, sometimes they don't. There is no good or bad. It depends, or maybe, as the Taoist farmer would say. So getting back to how can we use whole genome sequencing to assess health and wellness? One example is if a person's genome indicates that they're having a higher risk of developing heart disease, for instance, they can take steps and strategies by adopting a healthy diet, increasing their physical activity, monitoring their cholesterol levels, and reducing stress. A good example specifically in cardiology is that if we consider a patient with unexplained left ventricular hypertrophy, also called an enlargement of the muscle of the heart, presumed to be typical hypertrophic cardiomyopathy, actually about 1 in 50 of these patients will have a condition called Fabry's disease, which has a very specific therapy through enzyme replacement therapy, but unfortunately, this is often overlooked. Similarly, if a person's genome shows that they're at higher risk for developing certain cancers, we can work with their healthcare providers to develop a personalized screening strategy and surveillance plan to detect that cancer, or the cancer that they are at risk for, at an earlier stage when it's most treatable. This would mean performing screening tests on them that we wouldn't perform on the general population, and the goal would be to detect the cancer before it gets out of hand. Another example of whole genome sequencing used for health is it can be used to identify gene variants that may impact an individual's response to medications. This field of medicine is called pharmacogenomics. A patient with a genetic predisposition to diabetes may also be advised to make dietary changes to improve their exercise to prevent the onset of diabetes. Whole genome sequencing can also help individuals optimize their health and wellness by identifying gene variants that may affect traits such as metabolism or identify inborn errors of metabolism and advise their physical activity and their nutritional needs. Now, we are all born with variations that affect our health. On average, we have about 5 or 10 de novo new mutations in each of us, and this is just part of the evolution of life. Sometimes these turn out to be good for us, and sometimes they turn out to be bad for us. Whole genome sequencing is just information, but that information can empower us to take action and prevent disease. Armed with this information, individuals can make informed choices about their lifestyles and take proactive steps to improve their health and well-being. Now, I should pause and clarify that whole genome sequencing is not necessarily a diagnostic test. It's just information, and that information may prove useful. It's akin to a non-hypothesis scientific question. In contrast, if someone already has a suspicion for an underlying genetic cause of the disease, then whole genome sequencing might not be the best test to perform, and might want to do diagnostic genetic testing, specifically for the gene that is suspected to be the underlying cause. But whole genome sequencing is a helpful and useful screening tool that can't be relied upon for ruling in or out the disease, but it is still a good screening test. So meaning that the usefulness of whole genome sequencing is best as a screening tool, not necessarily as a diagnostic tool. But that said, it is a useful tool in the toolbox and is clinically useful, but can't be used in isolation. So typical primary care physicians sometimes don't realize how common genetic issues are in their patients that they treat. For instance, 1 in 17 people have a so-called rare disease, and 80% of these have a genetic or genomic basis for that disease, meaning that there is a significant number of patients in a typical primary care practice who have a rare disease and are often overlooked in the typical primary care setting. Oftentimes, typical primary care providers are suspicious of an underlying genetic cause only when they see so-called red flags, which are things not to be missed in their busy practice. For instance, if someone comes in with a family history of a condition with multiple family members affected, for instance, early onset breast cancer, they may start to think that there's something genetic going on, but oftentimes this is overlooked. Research says that about 5 or 10% of people who are diagnosed with cancer have an underlying genetic syndrome as the cause. But unfortunately, given the hectic nature of the healthcare system, with only 15 minutes with your primary care doctor, the underlying genetic causes are often overlooked, or there's just hand waving that happens that just says it must be due to your genes, but they fail to do anything about it. And I see that as a missed opportunity because we have amazing potential for interventions if only we were to explore that possibility. Primary care doctors are also, the typical primary care doctor is also not necessarily experts in managing the emotional consequences of harboring these genetic variations and in managing the anxiety or the reassurance that they need to help their patients assess their risk and what they can do to help themselves. One of the key roles that I play as a clinical geneticist is to assimilate all of this information, separate what is clinically relevant versus what is just interesting, and share this information to educate my patients so that we can translate those findings into real practice as quickly as possible. This is what I hope to offer you as a patient in my practice and how we are different than the typical medical practice. So in conclusion, whole genome sequencing has enormous potential to revolutionize healthcare by providing personalized insights into an individual's unique genetic makeup. By leveraging this technology, individuals can take proactive steps to prevent disease, optimize their health, and improve their overall well-being. If you're interested in learning more about my practice and how whole genome sequencing can benefit you, I encourage you to book a consultation with us today. We're located at Highland Park, Illinois, and you can find us online at www.harmonyinfocus.health.